guest lecturer for today. So, Dr. Lawrence Heaney is the Negoni Curator of Mammals in the Field Museum of Natural History. He took his PhD in Systematics and Ecology from the University of Kansas. He has been an adjunct curator of several museums in the country, such as the National Museum of Natural History here in the Philippines and University of the Philippines, Diliman. He is one of the foremost experts on Philippine mammals and Philippine biogeography and has published numerous working with different collaborators from the Philippines, such as Mr. Danilo Balete, the late Mr. Danilo Balete, Ms. Pipi Veluz, Mr. Philip Alviola, Dr. Leticia Fuang, who is also joining us, Mr. Aloy Duya, and other mammalogists and biologists in the country. His research interest is long-term evolutionary, ecological, and geological factors that influence patterns of biodiversity and endemisms in oceanic archipelagos. Once again, to our students, and our participants today, we are very lucky to have Dr. Lawrence Heaney as our research speaker for today. So once again, let us welcome Dr. Lawrence Heaney. Good evening, Dr. Larry.
Good evening to everyone. I'm honored to be invited to speak with you this evening. Hope we can have a good conversation. And I am delighted to see that quite a few of my illustrious colleagues who study mammals are going to be with us this evening also. So my hope is that we can turn this into a lively discussion. What I want to do first is run through a brief PowerPoint presentation. I will try not to talk for too long so that we can have time for a good discussion. So what I'm going to be doing this evening is, or evening my time, it's 8 o'clock in Chicago right now, is responding to the request to talk about to give an introduction to ecological studies of mammals in the Philippines. So I work on a variety of different kinds of issues, as you've just heard about. Some of it is biogeography, some is systematics and evolution, some things that are specifically conservation biology. What I'm going to focus on for now are ecological issues involving studies of Philippine mammals. So, Eric, if you can bring up that PowerPoint presentation, we can get started.
There you go.
Okay, can you go to the beginning? There we go. Okay. All right. So, yes, brief introduction. I'll try to keep it brief. Next slide, please. So one of the things that I want to point out at the outset is that one of the very best things you can do if you are interested in this topic is to attend some of the national meetings. There are two organizations that are very active. The Association of Systematic Biologists of the Philippines, and the other that is pictured here is the Biodiversity Conservation Society of the Philippines. I find that most of the people who I end up interacting with over the long run, I meet at these conferences. When we started off with the BCSP in 1992, I think we had 26 people who attended. Now we typically get 250 or 300 and sometimes more than that. So it's a very large and very, very lively crowd and a great bunch of people. So I strongly recommend that. It's a great way to learn and an awful lot of fun. Okay, next slide. So one of the first things that you need to be aware of is that the Philippines is one of the most geographically diverse areas in the world. We tend to think of it as just being a single archipelago of islands, but actually in terms of the biology, it's extremely diverse, and that has a very, a very great impact on the kinds of studies that you're likely to do depending on where you live and where you're doing the research. So the primary factor that determines this is something that may be familiar to many of you. At one time, it was thought that the Philippines had been connected by dry land to the Asian mainland. We now know that that is not the case. Instead, maximum distribution of dry land is what's shown in this map. So the current islands are shown in dark green. Areas that remained continuously as seawater are in blue. And that kind of pale green shows the extension of the lands that took place during the ice ages. The most recent one of which extended from about 18,000 years ago until about 12,000 years ago. So what you can see from this is that Palawan expanded out considerably, but didn't make contact with other islands, any of the larger islands. Luzon merged up with a couple of medium-sized islands, Marinduque and Catanduanes, for example, but did not make continuous contact with the islands to the south. Mindanao expanded out to include Leyte and Samar and Bohol and a number of other islands. In the Visayas, the western part of the Visayas, Heaney, Negros, Cebu, and Masbate all joined up into a single large island. So these are areas where animals were readily able to simply walk from one dryland area to another over periods of thousands of years. So if we go to the next slide, you can see the impact of this. Okay, so starting up in the upper corner with Luzon, the island of Greater Luzon that existed during this most recent period of low sea level. 90% of the mammals there occur nowhere else in the world, of the non-flying mammals. Bats are more widespread. They can fly between islands much more readily than a mouse can swim between islands. But 90% or perhaps more of the mammals that live on Greater Luzon occur nowhere else in the world, not even in the Philippines. Down in Greater Mindanao, again, 93% of the species down there don't occur anywhere else, including not on Luzon. Greater Negros Panay, 50%, nowhere else. Greater Mindoro, 75%, nowhere else. So each different part of the Philippines is a distinct center of mammalian diversity. So one of the first questions that you have to ask yourself is, well, where am I going to do my study? That will have a very, very big impact on what's available to you to study. Next slide, please. Another major factor that will have an impact on your ecological studies is the elevation at which you do your studies. So for most of you, I'm guessing you're going to be at relatively low elevation, so not above, excuse me, not above 1,000 meters. That means you're going to be in what at least was originally some form of lowland forest. Much of it, of course, has been quite disturbed by human activities now, but that was the original plant formation. That extends up to about 1,000 meters. For those people who are who live in Baguio, they live in an area that originally was covered by montane forest, a very, very different kind of plant formation. And still higher, where not very many people live, is mossy forest. As you go up in elevation, temperatures decline. So it's much cooler as the higher you go in the mountains. In addition, as you go up in elevation, rainfall increases on average. So, if you're in the lowlands, you probably are in a place that gets roughly 2 meters of rain per year. There are places up at high elevation that on average 5 meters of rain per year. These are very, very wet habitats. So the ecology of the area and the presence of the mammals is going to be very strongly influenced by the part of the country where you work and the elevation at which you work. Next slide. So just as an example of that, on the left, you see bats of the family Hippocideridae, the genus Hippociterus. These are really cool little bats, wonderful things. They eat insects and other invertebrates. So the graph that's above them shows the distribution of each of the species, in this case on Luzon Island, along the elevational gradient from sea level up to about the top of Mount Pulag at about 3,000 meters. And what you can see there is, of course, that most of the records are from the lowlands. Bats are most abundant and most diverse in the lowlands. With the non-flying small mammals, which are extremely diverse, lots of species. In the lowlands, there tend to be rather few species, but as you go up in elevation, the number of species increases. So again, the place that you choose to do your study will have a big impact on what is available for you to study. Next slide. All right, so some basic steps in deciding about your project. I'll try to go through these things fairly quickly. Some of them we'll come back to. All right, first, you need to decide what it is you wish to do. What is it that you want to learn about? So one of the things that I can tell you now, and I think that all my mammalogical colleagues who are here will agree that there is just a tremendous amount to be learned. Any place you live, any place you're going to be able to do field work, you will be able to do interesting things. You simply have to start by deciding, what are your objectives? What do you want to try to accomplish? What do you wish to learn? More than likely, what you learn, it will be the first time that anybody has learned those things. Okay, in terms of practicality, you need to very quickly decide whether you're going to study bats or study small mammals. Or you can choose to, of course, focus on a single species. Now, I'm not going to say anything more about the large mammals because they're generally very heavily hunted. They're hard to observe. It's hard to get information on them. It's a very specialized kind of thing. But that leaves the vast majority of Philippine mammals to be studied. We'll come back to this. Next thing, are you interested in trying to work in something that is close to natural habitat, either undisturbed or forest that has regenerated quite well into a natural state? Or are you interested in working in disturbed habitat to find more about the impact of human activity on mammals? Another thing that we've talked about here, which faunal region of the Philippines are you going to work in? That will have a very, very big impact on what you can do, what species you're going to be working with, and as pointed out in the next line, will guide you to the appropriate published literature that already exists. So, of course, at this point of your study, you need to do a review to find out what's been learned before so that you can build on the foundation that others have established already. Okay, next, you need to choose your methodology. How are you going to conduct your study? Is this something that, we'll talk about some of the possibilities for particularly comparing bats and small mammals. Before you get started, you need to make a list of the species that you think you may encounter and get information together on how you will identify them. This is fundamental before you go into the field. You want to know what you're most likely to encounter. Now, it's important to keep in mind that the rate at which we discover previously unknown species of mammals in the Philippines is still very, very high. And depending on where you're working, some places it's unlikely, other places it's very likely that you would encounter species that no scientist has ever seen before. So you need to make a list, you need to know how to identify things, but you need to keep an open mind about what you might encounter. And then finally, I'll just mention briefly at this point, of course, you absolutely must obtain the appropriate permits. Don't do field work without permits. You're going to end up in big trouble if you do. You don't want to do that. Okay, let's move on from there. We'll talk more about a bunch of these things. So next slide. All right, bats. The diversity of bats in the Philippines is fabulous. These drawings give you some idea of the range of appearances of these animals. They're fabulous. I want to point out that in the lower left-hand corner is a golden-crowned flying fox. By weight, so far as we know, that is the largest bat in the world. They weigh up to probably about 1,400 grams at times. That's a huge bat. They've got a wingspan of close to 1.5 meters. Now, immediately above it to the left, tiny little inset there, is one of the smallest bats in the world. That is a lesser flat-headed bamboo bat. The English name is bigger than the bat. They are so tiny that they can crawl inside, if there's a crack or a hole in the side of a piece of bamboo, they'll crawl inside and roost inside the hollow space in the bamboo. These are really tiny bats. Both of these bats are still actually quite poorly known. Any studies that you do that involve them will produce new publishable information, I promise. The others, well, they're extremely diverse. My guess is that quite a few of you might end up doing research on bats simply because they are abundant and diverse in the lowlands, and that's where you're most likely to be, I'm guessing, doing your research. Next slide. So, for studying bats, the ecology of bats, there are a number of different approaches that you can take. Mist nets are very heavily used. And I have behind me here the visual aid. Here is a mist net. You can see that this is a brand new one, hasn't been used yet. It's made up of extremely fine black threads, nylon threads. So you unfold this, and if Eric, if you can go back to that PowerPoint. The nets, then these are typically they have 4 levels. Most of the nets are 12 meters long, and when they're set up, they are about 2 meters high. You can set them up in a number of different configurations. Philip Albiola, who's with us this evening, is a master of using different arrangements to catch some bats. Mist nets are particularly good for catching the many species of fruit-eating bats and nectar-feeding bats, the family Pteropodidae. They do not use echolocation. Most bats worldwide use a form of sonar to find their way around at night. So they're creating high-pitched sounds that they produce vocally. Those sounds strike objects in front of them, and echo back to the bat, which hears them, and they are able to use that information to, in effect, see their environment, but they're doing it strictly with their ears rather than with their eyes. All right, so those are the bats. The ones that have echolocation are the ones that catch insects and other animals. The fruit bats don't have echolocation. They orient visually. Well, these mist nets are very hard to see, so they just blow into the nets. So it's easy to catch them, relatively speaking. and mist nets are the preferred way to do that. The insectivorous bats are harder to catch because they do have echolocation. They are often apparently able to detect the mist nets, so they're much harder to catch. You can catch them that way. There are other ways to catch them as well. But another way that is being used increasingly frequently are devices that are called bat detectors. And these are electronic devices that are able to detect and record the sounds that the insect-eating bats make when they are flying and when they're trying to catch their prey. And you can identify the species of bats by the characteristics of the call. So it's a little bit like identifying birds by their song. Only in this case, you can't hear the song. You need to have a bat detector to do that for you. There's good literature on this. You can check that out. Another way to study bats at times is simply to watch them. The big fruit bats, the giant flying foxes in particular, typically roost at the top of very large trees where they're very conspicuous, very easy to see. For those, all you need is a pair of binoculars and a notebook, and you can do quite a lot. And then another possibility, and this is one that Aloy Duya has used to some extent, is radio telemetry. You put a radio collar, a radio transmitter on a collar on a bat and let it go, and you then spend the next couple of days trying to figure out where it's gone, follow it around, get information about their movement patterns. Again, there's information about that, that methodology that's available. Okay, next slide. We're going to keep moving along pretty quickly here. Now, most of you, I gather, are in the Visayas or Mindanao. So I've included this, and by the way, I'd be happy for all of you to get a copy of this PowerPoint presentation. You're more than welcome to it. This is a field guide to the bats that we put together a number of years ago, specifically for Mindanao Island, but it works pretty well more broadly also. Our book from a couple of years ago about the mammals of Luzon Island also has information about each of the species and how to identify them. The bat faunas tend to be relatively similar throughout the Philippines. There are some local differences as well. At any rate, this is the kind of information that you can use to identify the bats from photographs, combination of photographs, and then the information about measurements and basic characteristics that are shown in this field guide. Next slide. Can we go? Yeah, there we go. Okay, now for small mammals, again, one of the ways is to do direct observation. So if you live on Leyte, for example, or in Mindanao, there are animals that are active during the daytime. Tree squirrels, pygmy squirrels. At night, there are tarsiers and flying lemurs around. So with a pair of binoculars, and if you're out at night, a flashlight, you can get a fair amount of direct information that way. Another way to work with small mammals is to trap them alive in a cage trap, mark the animal in some way, and release it so that you can recapture it later on. This is a standard procedure that is used throughout the world. It's a lot of work, but it can be very, very productive. There have been very few studies using this technology. As a result, in many respects, we know very, very little about the ecology of most Philippine small mammals. And we just lost the PowerPoint. Eric, can you bring that back up?
Can you already see the PowerPoint, Dr. Heaney?
Ah, there we go. There it is. Okay. All right. Again, radio telemetry. as something that can be used on small mammals. It's been done in a number of cases. One study that comes to mind involving tarsiers was extremely productive. And then finally, I'll mention also that in some instances, using what are called camera traps is very effective. So essentially, it's simply a camera that you put out in the forest with a motion detector that causes it to turn on when something passes in front of the camera. This can be a great way of getting information about a wide range of species, particularly the things that are somewhat larger. So civets and, well, tree squirrels are big enough to set them off. Some of the larger ground-living rodents, and occasionally you may get lucky and learn something about the wild pigs. Okay, so again, each one of these requires specialized efforts. It requires specialized equipment. You simply have to decide which one you're interested in and to some extent, which one is feasible for you. Next slide. Okay, so the diversity of animals that you may be working with in each different place is going to vary considerably. On Luzon Island as a whole, the species that you see here are represented, and in some cases there are even a few more that are being shown here because some of them look a fair bit alike, particularly animals that live in different parts of Luzon. If you're going to be successful with a project like this, you are going to need to become adept at identifying animals correctly. As part of your project, one of the basic things to do always is to document the animals that you catch. So when you catch them, you should take basic measurements, depending on the kind of animal. With bats, often it's the forearm that is a key measurement for identification. With the highly diverse native rodents, the small mammals, tail length and hind foot length and weight often are highly diagnostic, very helpful in identifying things. Taking photographs is very important. If you've got good photographs, that will, if you have trouble, then perhaps one of the experts will be able to help you identify your animals. I will mention also briefly that at times one of the best ways to document a study is to keep some voucher specimens. That means you select a small number of the individuals that you capture, you kill them humanely, you preserve them well, and you deposit them in a good research collection. That is kind of the gold standard for this. That means that anybody can then double check your identification and make sure that it's correct. There are a number of posters like this that our project has prepared over the years. We can try to make those available to you. Next slide. Here's an identification guide for the mammals, specifically of Mindanao Island. This would be very useful on the other islands that were included within Greater Mindanao during the last ice age, so Leyte and Samar and Bohol and so on. Again, very similar to that bat guide, basic information about what to look for, to identify which major group your animal belongs in, and then information about measurements that will help you to identify them. Next slide, please. Okay, so this is a list of some sources of further information, and particularly examples of studies that use these different techniques. You'll see a few names here that you recognize, I'm sure. Not enough space to include all of the relevant ones that my colleagues here this evening have published by any means. There's a lot of really great stuff. for basic information about what mammals occur on in which parts of the Philippines, that the website up at the top, the synopsis of Philippine mammals, is a good place to start. The book that's immediately below it, Wilson et al., has lots of good basic information about how to conduct ecological studies. Engel and Heaney, The Key to the Bats of the Philippines, is something that is somewhat out of date now, but is still very helpful. Danny Belete's master's thesis from 1997 is one of the very few mark and release studies of native Philippine mammals that's been published and would be a great model for you to follow. R.S. Reginaldo's studies up in Baguio are nice, simple, straightforward examples to follow, and so on for the rest of you. So you can take a look through that. I presume that all of you are familiar. If you're not, you should be, with Google Scholar. If you go to Google Scholar and type in Philippine mammals, you will get an enormous list of references. If you are more specific than that, if you type in the scientific name of a species, you'll get lots of references about that particular species. There is a good deal of information that you can find to help yourself get started. I think that is the last of my slides. Yes. So now we can open this up for discussion and comments. I would love to hear from my mammalogical colleagues who are here to help fill in some of the many gaps that have been left here.
So thank you, Dr. Heaney, for that short lecture. So, of course, I would like to inform our guests that we will be joined in this discussion by a few mammal experts and, of course, biodiversity experts in the Philippines. For my students and for the other participants, you may start typing in your question in the chat box. I would just like to introduce the people that will be joining us for this discussion. From the University of the Philippines, Los Baños, we have Dr. Leticia Afuang. Good morning, ma'am.
Hello, good morning.
So also again, from the University of the Philippines, Los Baños, once again, we will be joined by Dr. Philip Alviola. Good morning, sir.
Morning, bro. Morning. Morning, everyone. Great talk, Larry.
Thanks.
From the University of the Philippines, Diliman. So he will be first joining us today, Dr. Aloy Duya. Good morning, sir.
Hi, good morning, everyone.
And of course, from the National Museum of the Philippines, she has already been my guest in this web series, Miss Sweepy, Miss Bao, Mrs. Mama, no, Miss Sweepy Velus. Good morning, ma'am.
Morning. Morning, everyone.
So while we, once again, to the participants and to the students, you may start typing in your questions because most of it, just to be fair with most of the participants, because some of them are joining us from far-flung areas in the country, and their internet connection is not that good. So I hope we could settle with your questions in the chat box. But before I go to the questions, while we are waiting for those questions to come in, Sir Philip and Ma'am Sweepy knows this, but I will also ask Larry about this. The first Miss Universe question I asked the guests in my web series is, what was your inspiration on doing this when you were just starting, when you were younger? What was your inspiration on doing these kinds of research, Dr. Larry?
Well, I have always loved spending time outdoors in nature. And I can't explain why that's the case, but it absolutely is. I've always enjoyed being outdoors and looking at plants and the birds and the flowers, but I have always especially enjoyed seeing, interacting with, and learning about mammals. Again, I don't know why. There are people who really love birds. That's great. I like mammals. I began working in the Philippines specifically because it has Well, we now know the greatest concentration of unique mammalian diversity per square kilometer of any country. I think that's fabulous. I want to understand why.
Thank you, Dr. Heaney. I would also like to ask that, Sir Aloy, when you were also starting, what was your inspiration on doing these kinds of research?
Well, again, I actually started as a botanist. Surprise! I actually work on mosses. So mostly in my undergrad thesis is on sphagnum mosses. So I always go up in the forest, and I also love outdoors. And then later on, I realized when you see the plants, it's there already. So there's not much challenge. So I shifted to wildlife because once you when you see the wildlife, you have to chase it. You know, so there's this. So this is more challenging for me, and of course, being in the forest is really a good, the opportunity of seeing this forest, intact forest. It's really very inspiring, and it really pushes you to do conservation work. So one of the things really, I think most of the common denominator ng lahat ng mga wildlife biologists here in the Philippines is that they love the outdoors. They love going out in the forest. And that's really, once you get to see the forest, it's really, it will really push you to do conservation work.
Thank you, Sir Aloy. No? Ma'am Letty, one of the very few female herpetologists in the Philippines. What about you, Ma'am Letty? What was your inspiration? Pang Miss Universe, ma'am, malay nyo manalo kayo today.
I think it is in the blood. I heard from my brother when he was still alive that he saw my mother capturing a cobra from a snake hole. But I didn't see that. But even in high school, I've been collecting frogs already. So I really didn't know I'm going to be a herpetologist. But the main push there was, in fact, I had to balance between, Larry has been very generous. I was trained at the Field Museum, and he invited me to join the group of the Mammals. I would have loved that, but there was a need in our department. No one was holding and developing and improving the studies on herpetology. None. It was a dying course, no? And I took the challenge. I said, I'll try to revive that with the late, my professor, Pete Alviola, the father of Philip. And I told him, sir, let's revive herpetology. I'll start off with the lab, you take the lecture, and let's see, because we cannot move on without balancing mammals, birds, and herps. So I became the herpetologist of animal biology. By force!
So we're not actually, I just noticed, no, we are not just actually having a mammalogist and biodiversity reunion right here, but it's also a reunion of different generations of biologists, no? From the, I do not want to say older science, but from the mentors down to the students, no? So we have a good diversity of people, of experts right now that would like to, that will join us in this session. So before I again to go to the questions, no, I would like to ask also Dr. Heaney. So again, so that I will not ask this question later, but I'm very interested to ask Dr. Heaney is, what was your best experience of doing your fieldwork here in the country? Your best and worst, the problems that you faced when you were doing your fieldwork here in the country. You can be as honest as you can be, Dr. Heaney.
So best is really tough because there have been so many good ones. Lots of really wonderful field work, lots of beautiful, beautiful, beautiful countryside. I think that probably the one moment that stands out more than anything else was the day that we were working on Mount Banahao, and actually finishing up the field work there, getting ready to head back up to Manila. And there was a typhoon that was kind of heading in our general direction. So we only had a couple of days to finish up the field work. And one of those last mornings, my good friend and colleague, Danny Belete, came back into camp with this very strange look on his face. And he opened up his hand and he had a mouse. And he instantly knew when he took it out of his trap, and I knew instantly when I saw it, that I had never seen anything like that before. Just fabulous animal. Its whiskers were so long that they reached to its hind feet. It had a second set of whiskers that came from a bare patch at the back of its eye. The hind feet looked like my hands, grasping hind feet. This was the animal that we named Masaromi's gulantang. It's a tiny, tiny, tiny relative of the giant cloud rats. Fantastic animal. And seeing that for the first time was definitely one of the high points. Bad, yeah, so I mean, the bad experiences basically have been times when, you know, something got off track and we weren't able to get into the field. That's the worst of it. As long as I'm in the field, I'm happy.
So before I go to Ma'am Letty and Sir Aloy as well to ask, so let's go to the questions now. So here's a question from Miss Danica Velasco, a fresh grad of BS Biology of Batangas State University. So she would like to ask how we would know if we have the right sampling size of the mammals we wish to study.
Sorry, the right what?
Sampling size. Sampling size.
Sample size, yeah. As many as you can manage. You can never have too many. Yeah, so you need to, you know, be thinking about how you're going to analyze your data, how you're going to present it, and you need to have a big enough sample size that it's meaningful, and that's going to depend on the circumstances. There are with certainly the studies that you will read about that, well, many of us have been involved in producing. There's information in there about sample sizes and how much you can interpret based on what you have. You do the best you can.
Thank you, Dr. Heaney. Sir Aloy, would you like to add some more advice to that?
Yeah, I think in terms of, again, as Larry has mentioned, you know, it really depends. Actually, it also depends on the questions, the objective that you want to accomplish. That would actually really tell you how much sampling that you need to do. So for example, you have to have at least a minimum of for mist netting. At least you have to have a minimum of 90 net nights per vegetation type at least. But again, it goes back again to the to your objectives. But somehow, if you read all the materials, at least a minimum of 90 as usually the basic ones that you need to do.
Thank you, Sir Aloy. Speaking of objectives, because these actually, you know, writing the research objectives is one of the problems of many students in biology. Is because usually when they write their objectives, you know, what they are writing is the summary of what they will do in the field or what they will do as their experiment. Ma'am Letty, I would like to ask you, Ma'am Letty, what advice could you give our students on how to write properly their research objectives? Because we've already talked about that the sampling will always depend on their objectives. objectives. So, Ma'am Letty, any advice to our students on how they can properly write their research objectives?
Um, for research, basically, you have to determine the period of time that you can allocate, and put yourself in a position of setting up an objective that you can accomplish within that given period of time, and within the given available resources that you can have. No, for students, it doesn't have to be very complicated, and usually there can be a general objective and some specific objectives, and these specific objectives will be the ones that will actually lead to the actual methods that you are going to implement in your study. No, usually as a young or starting biologist, it will be very difficult as of yet to envision what you are going to be in the next years to come, right? What you are really looking at would be how are you going to finish the course, and how are you going to meet your requirements for your thesis, just like that. But I think it is actually the professor's role to guide the students of what could be accomplished within, let's say, a maximum of 1 year, if that should be the case. No, don't let them give them the privilege of being able to do a short but relevant and significant study within a limited period of time, but give them the inspiration to go beyond that. Like, let's say you start off with this, but it can grow into something beyond that later on, and then inspire them. Like, I'll connect you to Larry Heaney, I'll connect you to Philip Alviola, join the expeditions, go and join Aloy Duyas, do your research in Sierra Madre with Dr. Duya, and even with Dr. Afwan. Yeah, that's how you do it. No, we cannot really drown them in big things at the moment, but we can inspire them of what goes beyond their graduation day, and that will be the one that will let them move to bigger things. I go goosebumps. Anyway, that's it.
Yeah, but let us allow them to graduate first so that they can do other things. Dr. Heaney,
Yes.
back to the questions, your ideas and comments on this, on writing research objectives and planning your research better.
So I think that Letty's point about first relying on your instructor to give you good advice is the right place to start. It depends on what you're interested in doing. And there are just so many possibilities that it's hard for me to respond briefly. Do it thoroughly. Whatever you try to do it thoroughly, do it carefully. Collect as much information as you can. Don't expect to solve huge major problems with your very first project. A lot of ecological studies are almost purely descriptive. That's okay. That's good. That means you're generating new information, and we need new information about virtually every aspect of the ecology of virtually every Philippine mammal.
Thank you, Dr. Heaney. So going back to the question, so from Tse Chen Lim from Malaysia, many thanks for the presentation that covers many good and useful things. One question, had the Greater Palawan and Greater Sulu ever connected to Borneo in the past during the ice ages?
Those are excellent questions. So as best we can tell at this time, the answer is different for the two. For Greater Sulu, it probably not is the current answer. It looks as though those areas were uplifted pretty recently, geologically uplifted. And the fact that they are geologically very young helps to explain the fact that mammalian diversity there is pretty low. So species that occur on islands of similar size along the coast of Borneo, tree squirrels, for example, tree shrews, lots of other things that occur on relatively small islands right along the coast, don't occur in the Sulu. That suggests to me that they haven't had an opportunity to get there, which means they haven't been connected by dry land. Palawan is a different story. Virtually, Palawan has a very, very rich mammal fauna. Of the non-flying mammals that are there, virtually all of them have their closest relatives on Borneo. They're distinct species. They've been different for some time, but their relatives are in Borneo. So right now, best guess that we can make is that Palawan connected up to Borneo sometime in the relatively distant past, perhaps on the order of 500,000 years ago, something like that. But those are really interesting questions. And gee, I'd love to see somebody do some creative work on trying to address both of them.
Hopefully we will inspire some of our students to do that kind of work. So before I ask you this question, I think that Sir Philip Alviola have already answered this from the last webinar, but I also would like to ask him again. So Miles Eric from Batangas State University would like to ask, among the methods for studying bats, which one is the most cost-effective and probably most efficient? So which one will you prefer for an undergraduate taking on thesis on bats? Sir Philip?
Thank you for the question, Eric, from a student as well. Well, before going into your thesis or working on the different methods, first of all, you need to have the necessary skill set to work on those methods. You need to be, adept with working on mist nets. You know how to set up a mist net. You know how to work on the bat detector or the tunnel traps. That's the first thing that you have to do before doing the thesis, because it might end up your results underrepresented or much worse. There could be some false negatives. That will definitely prove garbage on your data. So first you need to do some training, probably, or join an expedition, or probably hire or get some services of well-seasoned field technicians. That will definitely help a lot. But going back to her question about which method is the most cost-effective, well, like Larry and Ala has said, depends on what you're studying. Well, if you're gonna study fruit bats, Alloy et al. Has already published a paper on using sub-canopy nets, those what we call Skynet or Langit, setting up nets way above along the canopy. Their data has shown that you're gonna capture a lot more bats in terms of numbers and diversity if you compare it with ground nets. Of course, it's a bit difficult to set up Skynet. But yeah, for fruit bats, simple mist netting will do. You'd have to be very, very creative on how to set them up on the ground, across the river, across creeks. You can use V-nets for insect bats. Yeah, so for fruit bats, it's pretty straightforward. For insectivorous bats, it's a whole new ball game. It's totally different. Probably the easiest thing to do when you're working on insect bats is find a cave where there are lots of bats there. You can be sure when you set up your nets in front of a few distance from the cave opening. It's going to be very, very sure you're going to capture lots of bats, several species, and you can do several research or topics on caves just by setting up nets in front of the opening. Well, if you're going to do some very complicated things or doing some diversity studies, of course, the use of bat detectors, which is a little bit difficult to do, especially for undergraduate, because bat detectors can be quite expensive, and you do need some training on working on bat detectors as well. So yeah, for cost effective, for fruit bats, mist netting straightforward. For insectivorous bats, if you can find a cave or you can find a tree hollow, do a species-specific studies like what Danny did in his undergraduate thesis on Megaderma. He just found a tree hole, studied there for 1 year, great data, man. It's really, you come up with great data. Yeah.
Thank you, Sir Philip. So before I go to go back to the questions, Dr. Heaney, your comments. If you have other comments.
That all sounded like great advice to me. I would wonder what Aloy would have to add about that, given that he's done an awful lot of work with bats also.
So let's go to Sir Aloy. Do you have other advice for the student?
Well, yeah, mostly it's, well, if you're going to study fruit bats, it's really mist netting is the best way because, and again, although putting up canopy nets is quite time consuming, but it's more rewarding. You only spend shorter time to get as much as diversity and abundance of fruit bats in a particular area if you have those canopy nets compared to just using ground nets. That's what we observed so far in our studies. So yeah, canopy nets is really, yeah, I think the best way to go. If you have shorter time, you have only one site, you have shorter time, and if you wanted to do to document the diversity and abundance of these fruit bats, setting up canopy nets is the best way to go actually.
Thank you, Sir Aloy. Dr. Heaney, a question from Dr. Altamirano from the University of the Philippines, Visayas. So, why is it that the degree of endemism among the mammals of the Negros Panay region seems to be lower, compared with Luzon and the other regions?
Yeah, so that gets us into the biogeography and evolutionary studies area rather than really strictly ecology. But yeah, it's a very interesting question. So I think that part of the answer to that is that some of the islands are quite young. I'm not sure where the latest data stand, but for Negros, for example, at one point, I was told by a very knowledgeable geologist that Negros Island, which is, it's a very large island, maybe as little as 250, maybe 300,000 years old. Very, very young. That's given the rate at which colonization takes place among non-flying mammals. That's not very long. Now, Negros and Panay, all of the islands have quite diverse bat faunas. Bats are able to fly, so they can move from one island to another fairly readily. The data that I showed you on those maps are for non-flying mammals, native mammals. I think that the islands are young enough that there hasn't been time. Now, another one of the issues that actually you touched on at the beginning is that there has been much less field work done on Panay than on the other relatively large islands, and particularly in the mountains there in the western part of Panay. So I think that probably at this point, we don't know of all of the species that are present there. And so those numbers may very well be, to at least some degree, underestimates.
Thank you, Dr. Heaney. This question is from Danica Velasco. No? And I would also like to ask Ma'am Sweetie Veluz about this question. So if I were given an opportunity in the future, I'm also interested to being a curator in an institution or organization. In connection to this, how can I collect animals, specifically mammals, without collecting too much but with meaningful size? What logistics should I accomplish in preparing and exhibiting such collections of these mammals? Ma'am Sweetie, what is the National Museum's policy on this?
Currently, the National Museum, we do not have a general guidelines or protocols for how many culture specimens or specimens that we can collect. Researchers still depend on the design of the project or the study. And usually, as of the moment, most researchers from the National Museum do general collecting. But then, for some who have with projects, researches being done, like Dr. Jesmos for Terrestrial, and then for Dr. Acebes for the marine mammals. They have different protocols, and again, as the other speakers have mentioned earlier, it depends on the circumstances and design or the objectives of your research. So we really have no standard on how many specimens we collect from the field. So it still depends on the research. As for the exhibit, what we usually, well, for me, what I usually refer to people doing the exhibits, the planning for the display materials. I usually recommend the ones that are extra. I mean, usually there are collections or there are specimens that have sometimes people just come and go to the museum and deposit specimens. Some of them have no data, but we still accept them. We use them. We use them for exhibits or for teaching materials sometimes. Yes. So what? Except for some who would donate, like there are some people that we meet from the field, and they would like to donate some specimens that they have previously from their collections, from their would I say ancestors, mga minana. Yes. So some of our display specimens are donations. Yes. And we do not actually collect just for exhibit purposes alone. That's it.
I think Ma'am Supi did have a good point there. We need to clarify that the museums do not really just collect for exhibits. Because I think some of the people, some of the participants might have a different understanding of what the museums or the natural history museums here in the country and in abroad do. They do not only exhibit, but also do researches on different aspects of biology or whatever the taxa that they are handling. So it's not only just the exhibit. Dr. Heaney, in the Field Museum, are there other logistics that you do, or are there is there such a thing as collecting too much in museum collection?
Oh, of course. So there are some cases where you encounter very large numbers of animals. So, for example, if you go to a cave, there may be hundreds, thousands, maybe tens of thousands of bats living in the cave. There would be absolutely no reason that I can imagine that you would take any more than a limited number of each species. It's easy to catch lots of them. You let most of them go. depending on what you're studying, you may mark them and try to recapture them later or something of the sort. But at any rate, in terms of keeping specimens to be deposited as part of the research collections, yes, under those kinds of circumstances, you have to be very, very selective. In other situations where, for example, you're setting traps to try to catch small mammals, the amount of work that's involved is vast. It's hard work. You have to set an awful lot of traps to catch not very many animals. So the way that we prevent over collecting is essentially taking off from what Sweetie said, is to do our sampling over a limited area over a limited period of time, with the idea that from a given locality, we don't ever catch and remove more animals than a pair of owls would catch in a month. We know that the animal populations are able to sustain that level of predation, so we are simply mimicking natural predation levels. When that's done, I don't know of any evidence that will harm natural populations. But you and Sweet Pea made very good points. Natural history museums typically have two different sides to them. There's the public education program, which includes the exhibits, but most natural history museums also are biodiversity research institutes. And so the research that's done and the collections that are built often are not intended for display. They are there as part of a research collection. At the Field Museum and some of the other large natural history museums in the US, that research component is a very large part of what the institution does overall.
Thank you, Dr. Heaney. Going back to the ecological diversity questions. So here is a question from one of my students, Jade Carey Palma, from University of the Philippines, Visayas. So she would like to ask, is there a specific number or a range of animals needed for doing mark and release capture methods when studying a specific population? Dr. Heaney, is there a specific range when you do mark recapture methods? Or is there a number?
Right. More is better. But there have been some very, very important papers published on, Philippine tarsiers, for example, in which only a few individuals were captured. In those cases, they were the ones that come to mind. There were radio transmitters placed on them so that they could then be followed around. It depends on what you're doing. There's not a minimum number, but the more you're able to capture and mark and release and then later on recapture, the more information you're going to be able to accumulate about how far do these animals move, how large is the home range, how many different individuals overlap their home ranges. Do adult females overlap with each other? Do adult males overlap with each other? When the young are weaned and leave their mother, how far do they move? So, for questions like that, even with a relatively small sample size, you may be able to show some fairly significant differences. But the more individuals you're able to work with, of course, the more you're going to be able to squeeze out of the data.
Thank you, Dr. Heaney. Another question from my student, and I think Sir Aloy can answer this first before Dr. Heaney could comment. You have mentioned about radio telemetry as one of the methods to monitor bats. However, radio telemetry can be a little bit challenging, most especially to those who are just starting in the field. What she would like to know is if it has ever been used in studies in the Philippines, and what could you recommend to do when using radio telemetry? Sir Aloy?
Yeah. Well, first of all, Well, radiotelemetry should be done, because it's quite expensive, and it's really very difficult to track the animals in the wild. So I think that's the least study. If you're starting if you're a starting researcher, I think that would be the least method that you will use. You have to, again, we always emphasize, like Philip mentioned, that you have to train yourself first before venturing to those kind of, because you need a lot of resources for that. Not just because, particularly in radio telemetry, not because the equipment is expensive, but you need a lot of manpower to do that, to have a good data. We did the telemetry work on Terrestrial, as mentioned by Dr. Heaney. Yeah, we have we tag about 10 individuals and track them continuously. So it's really, it's a challenging, work to do. So I would really recommend first to really train yourself first, and also understand the species that you wanted to work on. Because you have to have at least a basic knowledge of the biology and ecology of the species, so that you will be able to design your radio telemetry work in the field, appropriately. It will entail a lot of resources. That's why you have to think well about your research design if you venture to this kind of, although it can be done. We have done, even for in Palawan, we did a radio telemetry for fruit bats, at least 2 species of fruit bats. And what's interesting, we did one for the Tenocyrus jaguari, and then we did one for Haplonycteris fisheri. Both are at least forest dwellers. And it's interesting, Larry has been mentioning if there's an overlap between male and female. Somehow our initial data indicates that for Haplonycteris, it seems that females have a separate, concentration of feeding area than the males, those kind of initial things that we're finding right now. It can be done. Again, but you just have to first learn the method, understand the species that you wanted to do. And then again, what we did with the Terrestrial, the reason why we ventured to that was because we only work on a small area. This is about 36 hectares only. This is in Initao Protected Landscape and Seascape in Mindanao. So one of the reasons why we ventured to that telemetry, because we are confident that once we tag the individual, we can look for them in that area, because it's expensive. Once for radio telemetry, once the battery died down, it would be very difficult to look for it. Although there are some, like the GPS loggers that's being used right now, that you can actually automatically track them. And you have, there are several telemetry transmitters that are available right now, but they are really very, very expensive. So we have to be careful. We just don't jump on those kind of methods, because there are misnetting. You can still do, studies habitat use using misnets, because mostly telemetry it's either home range or habitat use. That's one of the major application of telemetry. But again, you first use the simple methods. Those are readily available. I think that would be the first step. And then if you want really to venture onto specific species like what we're doing right now for the Philippine tarsier, then you can really focus on their behavior, reproductive cycle, how they, their behavior during breeding, those kind of things. You can do that specifically for radio telemetry. But then again, use first the basics, the basic ones. That would be you learn how to handle. You learn field work. You gain experience in the field before you venture to those kind of research work.
Thank you, Sir Aloy. Dr. Heaney, your comments.
Yes. So I agree with Aloy that for most of your students, you need to start off doing something that's relatively simple. So if you're interested in working with bats, mist netting for fruit bats is the place to start. The mist nets are relatively inexpensive. The fruit bats are relatively easy to capture. You can get a lot of information relatively quickly. And you can do that in a wide range of different types of habitats. As you become more experienced, you can perhaps justify going to the more expensive techniques that allow you to address things more specifically. But my advice is try to start out as simply as you can. Do the things that you know will work and that will produce data, and build from there.
Thank you, Dr. Heaney. Another question from Nourainir Laden Gilal, a BS Biology student from Mindanao State University, General Santos City. This is a very basic question. Which of the methods would be likely to produce a productive output when I'm not sure what he means by a productive output when conducting a study on biodiversity of small mammals in Mindanao? Maybe Dr. Heaney, you could just give some advice on what kind of study and what kind of method could he do with the small mammals, a study on small mammals in Mindanao?
There are lots of options. So one example would be if there's a forested area, lay out some trails, get a pair of binoculars, and every day, starting at dawn, walk the trails and note down what you see and where you see it. Information on habitat use by tree squirrels and pygmy squirrels would be extremely interesting. If you can go out safely at night, go out with a flashlight and a bunch of batteries, because you're going to need them, and observe flying lemurs and tarsiers. We have little information about how individuals of those species use the habitats. I think that doing live trapping of the smaller mammals that are active at night and hard to see, mark and release would be very productive. One of the things that we haven't mentioned, by the way, is that with both bats and most of the mammals, they're easy to mark in a more or less permanent fashion by getting what's called ball chain, and you just put a necklace around the neck of the animal and put typically what I've done is to use the kinds of bands that are used for marking birds with a little number on the band, and use that to mark the individuals. You mark them, you release them, and you continue your trapping regime over the course of 4 days every month, something like that, and you see where you recapture them. Get basic information on movement patterns, find out how many of them occur per hectare. Those kinds of things are very, very productive and are badly needed at this point.
Thank you, Dr. Heaney. Another question from one of my students. I don't know if Sir Aloy and Sir Philip could also add to this. Hirokems Rupia, a BS Biology student from the University of the Philippines, Visayas, wonders if there's already a study on migrating bats in the Philippines. Sir Heaney, are there, do you know any study on migrating bats?
There are. The short answer is no. And it would be wonderful to know more. So there are a couple of things that I can mention. One is that we have some very limited evidence that some species of fruit bats may migrate up and down mountainsides over the course of a year, depending on the season. One of them that actually occurs on Luzon, Odotoropus, is the genus. At certain times of the year, you catch only males at one elevation and only females at the other elevation. Well, obviously, they've got to get together at some point. So it seems pretty clear that there's seasonal migration of some sort going on. Nobody has studied that yet. The free-tailed bats, the members of the family Mollusidae, are migratory in many places around the world. Some of them roost in caves, at times literally in the millions. Alloy has seen some of these up in northern Luzon. Massive numbers of bats. Well, there are reports of them being present at some times and absent at other times. Well, that suggested that they're migratory, but we don't know anything more than that. So yeah, it'd be a very, very interesting topic if you've got the right situation to be able to investigate this kind of thing.
Thank you, Dr. Heaney. Sir Philip, any plans with your group to do studies on migrating bats?
None at the moment. But there was a simple exploratory work that we've done in Polillo, wherein we attach a radio collar, real-time satellite feed, radio collar on an Acerodon jubatus. This was like 10 years ago, related to our bat virus work. And a few days after, 1,000 miles after, it was located off the island of Guam, the signal. So it's like about 1,000 kilometers from Polillo. And then the signal went off. So that's a Cerodon jubatus. Wow. I mean, we don't know what happened, but we didn't get the funding for that, but there's initial data. Well, it was just an email actually coming from my Japanese collaborator. So, these flying foxes are really long flyers, but we didn't expect it to fly, 1,000 kilometers. But definitely within Subic, they've been doing telemetry work there, and some have flown per night like up to 80 kilometers per night, and then going as far as flying for 8 nights, but still localized along the Bataan Peninsula. But if you're talking about migration similar to North America or in Central America, I'm not sure, Larry. I think we don't have that here in the Philippines.
Yeah, the only ones that I would wonder about are the molluscs.
The molluscs.
I really wonder about those.
Yeah, they're the ones that capable of known.
Yeah.
Sir Aloy, your comment on this? Any plans on doing migrating bats research?
Again, Larry and Philip already mentioned most of the studies that has been done here in the Philippines. But just I'd like to add also the observations about Autopteropus. Yeah, I did my master's thesis in Mount Banahaw. So again, that observation that certain time of the year, in higher elevations, mostly males, we catch only males. Lower elevation, females, and then certain at the mid elevation, sometime September, October, there's a mix of male and female in certain like 1,300, 1,400 meters elevation. There's a mix of male and female there. So it seems that, those are some signs of, movement patterns within along an elevation for this species. So in other species, there's still a lot of, this only shows that there's still a lot of work that can be done. The students can, choose a lot of topics. And any data that the student can produce is very valuable because we need all those, those important data to really understand the biology and ecology of this species.
Yes, Sir Aloy. A question from Mesela Cola from Central Mindanao University. From your experiences in the field, is it also advisable to consider indigenous methods in trapping terrestrial mammals? Since our local guys are also knowledgeable in capturing specific species of mammals. Dr. Heaney, have you done this in your fieldwork using indigenous methods?
Occasionally, we've received specimens from local people who use their own traditional traps. Those traps are virtually always kill traps, so they, there would be suitable for only certain kinds of studies. Usually, they're intended to remove pests from agricultural areas. But they can be very, very effective, extremely effective at times. So yeah, that can be a good source of specimens, a good source of information under certain kinds of circumstances.
Thank you, Dr. Heaney. Another question from Marvin J. Sarmiento. I would just like to ask if there has been studies done in the 4 endemic zoos of the Philippines in terms of skull morphology.
Sorry, I didn't catch that. Could you repeat the question, please?
If there have been studies done in the 4 endemic indigenous zoos of the Philippines in terms of skull morphology.
The 4 endemic, what?
Zoos, S-U-S, Dr. Heaney.
Ah, yes, pigs, okay.
In
um,
terms of morphology.
Morphology. So a little bit of that has been done by archaeologists who want to be able to identify the material from their archaeological study sites. I can't offhand think of anything that has gone beyond that. Sweepy, do you remember anybody going to the National Museum to work with your pig collection?
What's this? Thomas, from the French Museum.
Mm.
But
Mhm.
he only he studied the skulls.
Yeah. So I basically, there's been very, very little done on that. And it would be quite interesting to see the results of a well-done study.
Did Ma'am Sweetie, just to confirm, did Nina check?
Nina Kib.
Yeah.
Kibod.
Yeah. Did Nina Kibot check on the skulls of the Philippine pigs or just the other herbivores? No.
I'm trying to remember if she was able to.
So,
She was able to check on the skulls.
because we have another colleague who is working, who is in.
I am not. I can't remember if she was able to check on the pig skulls, but the other large skulls for the herbivores, yes.
Okay.
Yeah.
Thank you, Ma'am.
Deer.
Yeah,
The
Eric.
deer usually. I think I remembered she checked on the deer skulls.
Yes, Sir Aloy.
Yeah, I think Dr. William Oliver did a study on measuring the skulls of wild pigs. I'm just not sure if it was published, but I remember I came across I think thesis of a student thesis doing skull measurements of wild pigs in Mindoro. Yeah, I think I remember those discussion before, but I'm not sure if it was published. Yeah.
So,
Right. Oh, yeah.
I don't think it was published.
it was not published.
I don't think so, no.
There was Eric Maynard.
Sure.
I'm not. Yeah.
Yeah.
Eric Maynard, who visited the UPLB Museum of Natural History and did some examinations of our pig skulls. Some coming from Panay. There's even some specimens from the Sulu archipelago. So I'm not sure if it already came out, but he did some measurements and definitely took some photos. Yeah, Eric Maynard. I'm not. Where's he from? Smithsonian? Is it? No.
No,
No.
not sure where he is at this
Yeah.
point.
Right. Sure.
But
Right.
there are some attempts to do studies on I think larger mammals. Yes, Ma'am Letty.
Yeah, there is a publication, a published book of, I think, the wild pigs, and some parts of those chapters. One was written about the zoos of Mindoro, and then that was with my group, with Joff and Maynard, and then also the Panay ones were written by, I think, the group of Lisa had about 2 chapters there. So I looked for that title. It's a book actually, and it focused on wild pigs. Okay, and it included the Mindoro and the Panay Philippine groups.
So a few publications as well. So probably because I think it's already quite late in Dr. Heaney's area. So I'll ask this last question. So from Wilhelm Tan from University of the Philippines, Los Baños. I'm wondering because he heard that certain mammals like civets and macaques are said to be introduced to most parts of the Philippines. How true is this? And what would be the possible explanation of these introductions? Dr. Heaney, your comments?
Sure. So um, the information is not absolute, but certainly it's very consistent with the suggestion that the civets, the 2 species of civets that are widespread in the Philippines, Paradoxurus and vivera, and the macaques, were introduced. So there are 2 different kinds of evidence that suggest that. One is that genetically, the populations within the Philippines are virtually identical to what's in Borneo or parts of Borneo. And that is surprising because there is among the non-flying animals, there is virtually no sharing of species between Mindanao, Luzon, Negros Panay, any of that, and Borneo. The genetics suggest that those animals arrived in the Philippines very, very recently. Just exactly how recently, that doesn't say, but very recently. The archaeologists tell me that macaques and the civets appear in the archaeological record rather abruptly about 3,000 years ago. That suggests that, there's enough information from archaeological sites prior to 3,000 years ago that if they were there, we'd probably have some evidence of that, and there isn't any. So it looks as though they probably were introduced sometime roughly 3,000 years ago from Borneo and were transported through the Philippines then rather quickly. Now, it is possible that these animals maybe were native on Palawan and just not in the oceanic portions of the Philippines that had never had a land bridge connection. That's less clear. Part of the problem is there just hasn't been a whole lot of research done on this.
Thank you, Dr. Heaney. I apologize for this because the question coming from one of my students from Palawan, this would be the last question. So she would like to understand the status of research focusing on the pangolin endemic in Palawan. So aside from poaching, could Palawan's biodiversity status possibly be affected by the decline of the pangolin? Because she remembers as a child that she has seen the species casually walking in their farm. And that was the last time she had encountered one.
Yeah. So just to clarify, this was a question about the pangolin?
Yes, Dr. Heaney.
Okay. So yeah, apparently for a long, long time, pangolins were relatively common on Palawan, and we're pretty tolerant of agricultural disturbance as long as there was some natural habitat that remained where they presumably had their nests and so on. What seems to have changed in relatively recent times is that with the huge increase in economic level in China, there is now a very, very large market for pangolins for human consumption. Both the meat of the animals and also the scales, which are used in some aspect of traditional medicine. So there have been shipments of pangolins confiscated by the DNR on their way to China that consisted of thousands and thousands of pangolins. So this looks as though your students' observations are accurate, the numbers have plummeted, and that this is a consequence of illegal, massive overhunting.
Thank you, Dr. Heaney. So before we end this, because I've already asked Sir Philip and Monsivi about this, so let's go first to Sir Aloy. Now, what would be your advice to our students who wish to pursue a career in research and field biology? So we started with the Miss Universe question, let's end it with a Miss Universe question. Sir Aloy.
Well, I think that my advice would be read journals, read articles. That's the very basic. You have to keep on reading. That's one way to really to learn and understand. And it also gives you a lot of ideas of what are the topics that can be done and what are the gaps and what are the research that are simple research that can really be easily done for a budding researcher. I think that's one advice that I would really encourage all the students to keep on reading. And also not just, you have to be updated. So if there's recent publications, you have to keep on tracking all those information, because a lot of things right now we've started a lot of answering some of the previously questions. So having a recent one would also give you a more background on the kind of research that you would like to do.
Yeah. Thank you, Sir Aloy. Ma'am Letty, any advice to our budding field biologists in the crowd?
I would agree with Aloy. Being updated is very, very good. But also try to be, try to join conferences. Try to join assemblies that discuss these things, or even webinars. Because in here, you get the first-hand experience of encountering the people who are actually doing the field work. You get inspired by, especially, surprisingly, these great scientists are very nice, very kind, very approachable, basically simple and friendly people, but they have a lot of things in their mind and in their experiences, and just talking with them and getting the feel of how they do it. You don't have to be very, very super intelligent at the very beginning. It will come on, it will develop as you focus on something that you love to do, find your passion, and give your heart to it. And definitely it will grow, it will flower, it will bloom, and it will produce a lot of fruits. Start simple. And yes, be teachable, be humble, know your limits, and look up to your mentors or teachers. They have a heart to inspire you, and we need a lot of scientists coming after us. We are the older generation. We need more young generations coming. Yeah, we are Philippine wildlife, Philippine biodiversity is very rich. We want much of our youth in there working with us.
Thank you, Ma'am Letty. So Dr. Heaney, to close this session, your advice to our students and other participants for those who wish to pursue careers in field biology and biodiversity studies.
So I would agree completely with Aloy and Letty. Doing a lot of reading in the scientific journals is really important, and typically is quite enjoyable. Just relax and enjoy it. But yeah, pursue it very actively. Both Philippine journals and foreign journals. A lot of good stuff coming out. I agree with Letty completely. Going to conferences, getting to know people is a really excellent way to learn a great deal. To that, I would add, get into the field, get outdoors. Almost doesn't matter where you go, but go somewhere. Get outside. Doesn't have to be in perfect habitat. Just be out there and just look, observe, dig in the dirt, chew on leaves, figure out what the bark on the tree tastes like. Be out there at different times of the day to see what kinds of activity is going on. Be a naturalist. Those activities are often what will provide much of the inspiration through your career. For me, the great majority of my good ideas have occurred as a result of doing field work, of spending time outdoors, just looking around.
Thank you, Dr. Heaney. So once again, thank you to Dr. Heaney for giving us this time and opportunity to talk with him and ask questions about mammal ecology. I hope he's not that we did not bother him too much this time because it's already probably around 10 or 11 o'clock in your side. So I hope all have learned a lot and of course enjoyed this session. So this will be my last episode for this web series. I need to rest and do other things, for this web series. Thank you as well to Ma'am Letty, Sir Philip, Sir Aloy, and Ma'am Sweetie for dragging you into this discussion. And hopefully you have also enjoyed the short discussion with Dr. Heaney. And I hope I also provided an opportunity for the mammalogists in the Philippines and as well as those that have been trained by Dr. Heaney to be reunited once again, at least for this very short conference. No, once in a while we need to see each other as well. Not even science. So hopefully,
Cheers, Eric. Picture.
ma'am, of course. So once again, thank you so much, Dr. Heaney, and to the other panel of experts.